Very good evening to all of you present over here. Myself, I'm Dr. Violet, and today we are going to start with this CSI NET question discussion, as already you all are part of it. And today we are going to discuss the questions from Unit 7, that is System Physiology Animals. So the questions will be from nervous system as well as from sensory organs. So before I start, please confirm if I am audible, if I'm loud and clear, if the slides are visible. Yes. Okay. So let's get started with the first question. So this question is from nervous system. Yes. So what is the question? When a nerve fiber is stimulated with increasing strength of stimulus. Yes, good evening students. Good evening Reshma. So we are starting with the first question now. So as I said, it is from nervous system. So it is basically a statement based questions. So today we are going to concentrate more on statement based questions, because if you see the nervous system questions, they are usually statement based questions. Okay. So when a nerve fiber is stimulated with increasing strength of stimulus, the action potential fails to generate even though the threshold level may be passed. The following statements may explain this accommodation of nerve fiber. A. The critical number of open sodium channels required to trigger the action potential may never be attained due to slow depolarization. B. Potassium channels open in response to slow depolarization, which makes the nerve fiber refractory to depolarization. C. The low threshold sodium channels remain open, which increases the threshold of firing of action potential. D. The efflux of sodium and influx of potassium due to operation of sodium-potassium ATPase oppose the depolarization. So you have to tell which one of the following is correct. Okay, I'll give you all some time. You can try and tell me, even if you're not very sure of it, you should try these type of questions, because now it is the practice session. So it will help you to understand that why my answer is correct or why my answer is not correct, right? Yes. So what do you all think? What should be the right answer over here for question number 1? What it depends on, whenever action potential is generated. So on what it depends. So the question is based related to that. Okay, so the graph of action potential is something like this, right? This is the depolarization, this is the repolarization, and then there will be a hyperpolarization state where it will become more negative and then return back. So it says a nerve fiber is stimulated with the increasing strength of stimulus. Even though it's increasing strength, the action potential fails to generate, even though the threshold level may be passed. Okay, so even if the threshold level is reached, still the action potential is not reached. Okay, so this is based on accommodation of nerve fiber. So it is related to the number of sodium channels, potassium channels, right? So let's discuss the answer for this question. The answer for this question will be option 2, students. Yes, Ashwarya, very good. Correct. You are right. Absolutely right. So that is how we try to solve these questions. You have to think properly, you have to analyze it, and you have to answer. So here, what it says, why A and B is correct. It says the critical number of open sodium channels required to trigger the action potential may never be attained. So here what is happening? Here the sodium channels are opening in the depolarization phase, right? Now you need some level of sodium channels, right? Some critical level. Otherwise, it will not give rise to action potential. Okay, you need some amount of sodium channel. So that critical level has to be reached. Okay, so the critical number of open sodium channels is required to trigger the action potential, and it may never be attained if there's a slow depolarization. If this depolarization is less, very slowly it is occurring, means that that many number of sodium channels are not there, right? So it will not be able to generate the action, this depolarization phase. So further, no action potential. So that is why A is correct, right? And B says potassium channels open in response to slow depolarization, which make the nerve fiber refractory to depolarization. So what is refractory? Refractory means it is a time period when one action potential is generated, so another cannot be generated. Okay, but here what is happening? Potassium channels have opened up because of this slow depolarization. Now, to generate another action potential, they are in the refractory phase, so they have to again go back, right? They have to uh, go back to the resting state. Clear? So that is why the option B is also correct. Potassium channels, they open in response to slow depolarization, so that will make the nerve fiber refractory. So that time another depolarization will not occur. Okay, so that is another reason why action potential is not being generated. Clear? So A and B will be the right answer over here. Okay, now let's see the C one. What does the C say? C says the low threshold sodium channels remain open, which increases the threshold of firing of action potential. Now, it is nothing like low threshold, right? Sodium channels remain open. If the sodium channels are open, if a critical number is reached, then definitely action potential will be generated. So this does not make sense at all. Low threshold sodium channel. There is nothing like low threshold, right? Threshold is the voltage that has to be reached, right? It is not about the channel, threshold channels. It is about the voltage that has to be reached. The threshold value has to reach. Okay, so C is again incorrect. Now D says the efflux of sodium and influx of potassium. So sodium moves out, potassium moves in, right? During the operation of sodium potassium ATPase oppose the depolarization. Now sodium potassium ATPase is completely a different no pump. It is a sodium potassium pump. So that does not play a role over here. This sodium potassium pump will play a role in maintaining the positive and negative ions, right? Inside it makes the negative and outside it makes it positive. But this, the influx of sodium and the potassium that is taking place, for them the channels are completely different, right? For this depolarization, you have just the sodium channel. For repolarization, you just have the potassium channel. So these are different channels. Clear? I'm sure now you all got it why D is incorrect. Okay, so D does not play a role over here. The sodium potassium ATPase will not play a role in generating the action potential. It will play a role in maintaining the inside negative of a cell and outside positive. Okay, the resting membrane potential that we have in the neurons of around minus 70 millivolt, negative inside. Clear? So the right answer for this question will be A and B. Okay, C and D are the incorrect statements over here. So I have mentioned if depolarization is slow enough, so the critical number of open sodium channels that is required to trigger the action potential will never be attained. So you need a particular critical number of sodium channels. Okay. Therefore, when a nerve fiber is stimulated with increasing strength of stimulus, the action potential fails to generate. Okay, even though the threshold level may be passed. So you need some critical level of open sodium channels to trigger the action potential over here. Okay, that is when when a particular number of sodium channels open, sodium enters inside becomes positive, so from negative it goes towards positive. That is when depolarization will occur, and that is when it will trigger that, give rise to the action potential. So you need some critical level of sodium channels that have to open. Right, now let's move on and see further. So I'm sure everybody is now understanding how to solve this type of statement based questions. Next is question number 2. It says a mechanical pressure was exerted on a specific location of a peripheral nerve of a mammal. The touch or pain receptors were stimulated from the skin surface innervated by the same nerve. The action potential generated by touch receptor stimulation was blocked beyond the point of mechanical pressure. But the action potential generated by pain receptor stimulation passes through the point of mechanical pressure. So following explanations were offered for these observations, right? So this question is basically based on the characteristic of nerve fibers. So here you need to know the nerve fibers. So there are three different types of nerve fibers, A, B and C. They have been characterized. So let's see the statements. A says the large diameter A fibers were affected by mechanical pressure. B statement is the small diameter C fibers were not affected by mechanical pressure. C, the intermediate diameter B fibers were affected by mechanical pressure. D, the large diameter A fibers were not affected by mechanical pressure. So here you need to know these characteristics of these fibers. A is what A is large or small, whether it is, no, it can be affected by mechanical pressure, by vibration. Okay, so these are the mechanical pressure means vibration, touch, pressure, pain, like that. So you need to know the characteristic of these A, B, and C fibers. If you know that, you can solve this question. So just try which and tell me which of the above combinations is correct. I'll give you all some time because you need to read thoroughly and try to answer. If you all know the characteristic, it will be easy to solve. If you have studied about the nerve fibers A, B, and C. Okay, Ashwarya, very good. Ashwarya has come prepared. Very good. Good preparation done for the upcoming exams. Others? You can try. See, it's very, it's like you will get a hint over here. If you know one statement, you will know that the other will be wrong. So in that way, it becomes little easy. Though it looks like it is little complicated. So you just have to know the characteristic of A, B, C. And in the question itself, they have given two very contradictory statements. So if you know one, you can easily know that the other statement is incorrect and you can get the right option over here. Reshma, good. Excellent. See, only if you try, then only you will be able to solve the question. If you just ignore seeing that it is very complicated, then you will never be able to solve, right? So you have to read each and every statement and you have to try to answer. Okay, so let's see how to solve this particular question. So as I said, you need to know the characteristic of A, B, and C. Okay, now if you know at least of A, you can easily find it out. So the first statement says Okay, so first let me tell the right answer for this question. The right answer is 1. Okay, the statement 1 is the correct. The option 1 is the correct answer over here. Okay, that is A and B. Now let's see how to solve this question. The first statement is the large diameter A fibers were affected by mechanical pressure. Now the A fiber, as I said, you need to know their characteristic. So A is having a large diameter, so that is correct. And they are affected by touch, okay, vibration, change, pressure, right? So that is why A is correct. Now if you know that A is correct, okay, so you can easily omit D because D is just contradictory to A, right? So the D statement says the large diameter A fibers were not affected by mechanical pressure. So if you know that it is A is correct, then definitely D is not correct. So where A and D, like you have to see those options, right? Even if you don't know the others. So you have to see where A is present and D is not present, right? So in that way, you will easily get the right answer over here. So the right answer will be option 1, right? So the option 1 is A and B. So A is correct, D is not correct. Now let's see B. The B says the small diameter C fibers were not affected by mechanical pressure. Now C fibers, let me tell you all the characteristic of C fibers. They are definitely small diameters, okay? And they are, when they have said that they are not affected, so actually they are small diameter. They are the smallest over here. They get affected by mechanical pressure, but it is very less. Okay, so it is not their major characteristic. So that is why this statement will be correct over here. They were not affected by mechanical pressure because uh, they will be slightly affected. Okay, it's not their major characteristic over here. That is why B statement will be correct. Are you getting it? Okay, so the right statements are, or the correct statements are A and B. D is incorrect, I told you. Now let's talk about C. Intermediate diameter B fibers were affected. So these B fibers are definitely not affected by mechanical pressure. Okay, so C is incorrect. Right? Got it? How to solve this particular question? So the right answer is option 1. So as many times you will see, in previous year question papers, they'll ask you questions from A, B, and C fibers. So this is very important topic from exam point of view. Okay, once they had, okay, Ashwarya, clear, right? So once they had even asked a question, but the question was actually little dicey. The statements were not very appropriate. So I think they had given the benefit of doubt over there. Whoever had solved, they were able to score the marks because similar question they had asked, but the statements was little not very correct statements they had written. So they had to give the marks. But this is important topic from where they have asked questions in the past many times. Okay, so let's see. So I'll quickly go through whatever is written over here first. So the simplest sensory receptors are free nerve endings that respond to bending or stretching of the sensory neuron membrane. So these receptors are known as nociceptors. So they are normally insensitive to mechanical and heat stimulation. Nociceptive pain is more common type of pain and is experienced by, this kind of pain is due to activation of small diameter afferent nerve fibers. Okay, so this is about the receptors. Okay, these sensory receptors, the nerve endings that can respond to different types of like bending, stretching, okay, the mechanical changes that occur. So the large diameter A fibers were affected by mechanical pressure, while the small diameter C fibers were not affected by mechanical pressure. Okay, so the large diameter sensory nerve fibers are the ones that mediate touch and pressure sense, or pressure, touch and pressure changes also you can say. Okay, let's move ahead and see the next question. So again, this question is from nervous system. Let's see what is the question over here. An action potential was generated on a nerve fiber by a threshold electrical stimulus. Means in this question, they have already said that there is a threshold electrical stimulus. Now, action potential is already generated over here. The question is, already action potential is generated. When a second stimulus was applied, no matter how strong it was, during the absolute refractory period of the action potential, the nerve fiber was unable to generate second action potential. So this question is a little different from the first one that we saw, right? Here, they're saying already one action potential is generated. Now, the second stimulus is applied. Okay, so during the absolute refractory period of the action potential, so that time another action potential cannot be generated. So the observation was explained in the following statements. A, large fraction of potassium channels was voltage inactivated. B, the critical number of sodium channels required to produce an action potential could not be recruited. C, a large fraction of sodium channel was voltage inactivated. D, the critical number of potassium channels required to produce an action potential could not be recruited. So which one of the following is true? Can you all try and tell me? So here the question is different. Okay, here already one action potential is generated. So you need another action potential over here. Okay. Okay, Anupam, good. But that is only one statement. Okay, there is another statement that you need to find out. Yes, Anupam, good. Right. Very good. Yes, Ashwarya. Correct. Yes. Okay. Okay, Ruben, good. So let's see this particular question. So in this case, it is already said, okay, that it is an absolute refractory period. Now always remember, whenever it is an absolute refractory period, even though the stimulus strength is very high, another action potential cannot be generated because they have already mentioned it is an absolute refractory period. Okay, it cannot be generated. Right. So here, the right answer for this question number 3 will be option 4. Okay, C and D. Okay, why C? Let's see the C first. A large fraction of sodium channel was voltage inactivated. Now, what happens is whenever another action potential has to be generated. So, I have told you all that there are sodium channels and there are potassium channels, right? But sodium channels have two gates. Okay, so one is the activation gate, another one is the inactivation gate. Activation gate, inactivation gate. So, that, like that, there are two gates over here. But potassium has just one channel. Now, for another action potential to be generated, the sodium has to return back to its resting state. So that is what it means over here. See, a large fraction of sodium channel was voltage inactivated. Means they are still in the inactivated state. They have not returned back to their resting state. Yes, Reshma, right. So C is correct. Okay, a large fraction of sodium channel was voltage inactivated. So they have to return back to their resting state. They are in the inactivated state. To generate another action potential, to go to that, after absolute refractory period, you have a relative refractory period. Okay, to go to that relative refractory period, the sodium channel, they have to go back to their resting state. Otherwise, they are still in the inactivated state. Okay, that is why C is correct over here. Now, A, what it says, a large fraction of potassium channel was voltage inactivated. Now, first thing is the sodium has to return back to its resting state. So, C is correct. And the potassium channels, okay, they also have to close, right? They have to close and that critical number of potassium channels required to produce an action potential has not yet reached. So potassium channels have to close, only then another action potential will be generated, right? And the sodium has to return back to some, in that relative refractory period, sodium has to return back to resting state and potassium channels have to close some critical level of potassium channels. Only then another action potential will be generated, right? So that is why C and D will be the correct options, right? What A says, a large fraction of potassium channel was voltage inactivated. So it is not about voltage inactivated. You need a critical level of potassium channels, as we were seeing in the beginning. When you have to, when action potential has to be generated, you need critical level of sodium channel. Similarly, so A is incorrect over here. B, the critical number of sodium channels required to produce an action potential could not be recruited. So this statement will not be correct over here, because here you're talking about already one action potential is generated and you need to generate another one. For that, the sodium has to return back to its resting state, because it is in the which phase it is in the absolute refractory period. Okay, so that is how we solve this particular question. So I'll quickly go through that what is absolute refractory period and relative refractory period. So absolute refractory period is the time during which another stimulus given to the neuron, no matter how strong the stimulus is, it will not lead to a second action potential. Thus, because sodium channels are inactivated during this time, additional depolarizing stimuli do not lead to new action potentials, because sodium channels are inactivated. They have to return back to the resting state, remember. Relative refractory period immediately follows the absolute refractory period, as voltage gated potassium channels open to terminate the action potential by repolarizing the membrane. The potassium conductance of the membrane increases dramatically. Potassium ions moving out of the cell bring the membrane potential close to the equilibrium potential. So you need a critical level of potassium channels over here. Okay, that will produce an action potential, because the membrane potential has to try to go back to the negative value. So potassium channels will open up, potassium has to move out, right? All those voltage changes have to take place. So that is why you need a critical number of potassium channels. So C and D will be the right answer, correct option. Okay, the option number 4. Okay, so let's move ahead and see the next question. Okay, so this particular question is again from nervous system. Let's see how, again, statement based question. A patient has episodes of painful spontaneous muscles contraction, followed by periods of paralysis of the affected muscles. It was identified as primary hyperkalemic paralysis, that is an inherited disorder. The possible causes of the paralysis are: the elevation of extracellular potassium causes hyperpolarization of skeletal muscle cells. B, the hyperpolarization of muscle cell membrane inactivates sodium channels. C, the elevation of extracellular potassium causes depolarization of skeletal muscle cells. D, the sodium channels are voltage inactivated in depolarized state. So which one of the following is true? So can you all try and tell me what should be the right answer over here? So what is this hyperkalemic paralysis? It is basically, the muscles become weak. Okay, more of potassium. So that is hyperkalemic. Okay, some students have already answered. Question number 4. Okay, Vanishri, very good. That is correct. Anupam, Ruben, I think you need to try again if you have answered for this particular question. Vanishri is right. Okay, let's see. Yes, actually, let's see this particular question. Ashwarya, we need to solve it. It is not correct. The right answer for this question number 4 will be option 4. Okay. So here, the possible causes of the paralysis. So first thing, okay, accumulation of potassium. So hyperkalemia means high level of potassium in the serum. Okay. So in the interstitium of the skeletal muscle, during repetitive action potential, depolarizes membrane potential and contribute to muscle fatigue. So too much of potassium will lead to muscle fatigue. Okay, so if the potassium levels are very high outside, so what will happen? It will depolarize the membrane potential. So it will lead to inactivation of voltage gated sodium channel that leads to the muscle fatigue. Okay, so this T tubule is basically the muscle fibers that are present over there. So the high potassium concentration in T tubule will depolarize the membrane potential. So if potassium levels are very high, now what happens? When we talk about the depolarization, repolarization, during repolarization, the potassium has to move out, right? Now there is more and more of potassium outside. Okay, so because of that, it will lead to the muscle fatigue. Okay, so it will depolarize the membrane potential, which leads to inactivation of this voltage gated sodium channel. So when depolarization will occur because of too much concentration of potassium outside, the inactivation of sodium channels will take place. Okay, so sodium channels are in the inactivated state. They will not be able to return back to the resting state. So that is why there will be a muscle fatigue that takes place over here. Okay, so that is the right reason why the hyperkalemic paralysis occurs. Okay, so the elevation of extracellular potassium will cause depolarization of skeletal muscle cells. So that will lead to sodium channels are voltage inactivated in depolarized state because they are in this state, right? They are in the depolarized state. Now, to generate another action potential, they have to go back to the resting state. So that cannot occur. So that is why the muscles will be paralyzed over here. Okay, they cannot move, right? The muscles become stiff, they cannot move. Clear? So that is why C and D will be the right options over here. The elevation, the A says the elevation of extracellular potassium causes hyperpolarization of skeletal muscle cells. So this elevation of this high level of potassium will not cause hyperpolarization. It will lead to depolarization because potassium will be outside, it will be more, right? So because of which there will be a change in membrane potential. So that causes this hyperkalemic paralysis. So A is incorrect. B, the hyperpolarization of muscle cell membrane will inactivate the sodium channel. It is not the hyperpolarization, it is the depolarization that will inactivate the sodium channels. Clear? And they will be in the inactivated state because of this depolarized state. Clear for everyone? Okay. So C and D will be the right options over here. So what happens when there is hyperkalemia or there is more of potassium? So in that case, there will be membrane depolarization. Okay, those T tubules over there will depolarize. So it will lead to inactivation of voltage gated sodium channels. So that causes the muscle fatigue. Okay. Now, before I move ahead, I just wanted to share with you all about the Raftar batch that is starting on this 11th September. So it is a batch where quickly all the topics will be covered, the very important topics, and it is like a speed revision. So whoever is interested, you all can join this batch. So in a very short time, all the important topics you can study and question solving will also be there. And you can also pass on this information to your friends, your juniors, your seniors, whoever is interested to prepare in very less time before the exam. And along with it, I also wanted to share about the exclusive cloning vector and recombinant DNA hands-on training. So anyone can attend it. It is online as well as offline. If offline, you can get trained at IISc incubated lab as well. So this is on 9th September. That will be starting at 10 a.m. So if you want to register, just call on this toll-free number 1800-1200-1818, or you can even get more help on 080-5099-7000. Okay, let's move ahead. Okay, Ashwarya, got it. Very good. Let's see the next question. So, okay, so now we come to sensory organs. Now, in recent, one or the previous years, I'm not seeing much of questions from years, but previously they used to ask, if you see check the old question papers, they used to ask question from ears. So ears and eyes, they are important, okay, from sensory organs, very important. So let's see some questions. The stereocilia of auditory hair cells are arranged in rows, but the height of stereocilia are not the same in all rows. Though the height of stereocilia is the same within a particular row, the heights increase in subsequent rows. Okay, so I know if you have studied about the hair cells, so there are 3 rows. Okay, you have outer hair cells, you have inner hair cells. Right? So you need to know that part of it. So when the stereocilia of shorter rows are mechanically pushed toward the taller rows, so you have like this, okay, some short hairs and then the tall hairs like this. So they are in this manner. The hair cells are depolarized. So what it says, when the shorter ones are pushed towards the taller ones, it gets depolarized. But when they are pushed away from the taller ones, they get hyperpolarized. Okay, so this direction will be depolarized and this direction is hyper. So already this information is given in the question. Now the question is the significance of this graded height of stereocilia. So why is this graded height? From small to little bigger and then it becomes more tall and taller like that. So this is a graded height. That is what it means over here. Graded height of stereocilia is. So you have to tell the significance why it is like that. Each row of stereocilia may be displaced independent of other rows in physiological conditions. B, the tip of the taller stereocilia will show greater displacement as compared to shorter ones when all the rows are moving in same axis. C, the hair cells will be depolarized or hyperpolarized in different grades when the axis of displacement is changed. D, the taller stereocilia are involved with depolarization and shorter ones are responsible for hyperpolarization. So you have to try to find out which one is the correct statement over here. Okay, just read carefully. Already much of the information is given in the question. Okay, Anupam, Ashwarya, very quick, good. But no, that the answers are not correct. You need to again check. Try again. You have to find the correct statements. Why you all have said option 4? There the D if you read. Okay, D, do you all think it is correct? The taller stereocilia involved with depolarization and shorter ones responsible for hyperpolarization. Is it like that? Now only we read, right? It is not like that. They are all moving together. Okay, not that tall one is for depolarization, short one is for hyperpolarization. Right? It is mentioned in the question. What is causing depolarization and what is causing hyperpolarization. So definitely D is not correct, students, if you read this carefully and if you understand how this movement is occurring. Okay, so anything else, any other option that you think is appropriate? Okay, very good, Ashwarya, good try. Yes, that is correct. Vanishri, try again. That is not appropriate. Ruben, try again. See, D is not correct. Okay, so D is not correct. Okay, but you have to go through each and every statement over here because it is not very easy options that they have given. So each row of stereocilia may be displaced independent of other rows. Will it happen like that? No, right? So they are arranged in rows, but the height of stereocilia are not the same in all rows. Though the height of stereocilia is the same within a particular row, the heights increase in subsequent rows. So when stereocilia, so this is how depolarization, repolarization is occurring, they have mentioned. So each row of stereocilia may be displaced independent of other rows. It will not be like that. Okay. So you have 3 different rows that will be there. So it will not be independent of each other. Okay. So all will move in the same direction. It will not, one will move like this, one will here, it will move like that. No, right? So they will not be displaced independent of other rows. So that is why A will not be correct, students. Okay. So if A is not correct, then this 4 is also not correct. Now let's see B and C. The tip of the taller stereocilia will show greater displacement as compared to shorter ones when all the rows are moving in the same axis. So it is not like that. Okay, it is not that this tall one will move till here and the short one will move only this much. It is not like that. Okay, so the tip of taller stereocilia will show greater displacement as compared to shorter ones when all are moving in the same axis. So that is incorrect statement over here. Sorry, what I said. The tip of taller stereocilia will show greater displacement as compared to shorter ones when all the rows are moving in the same axis. Okay, so what it means over here is, see, these when they move, okay, so when these short ones are moving, so first we know these short ones, they are in the graded height. Okay, so they will move. Okay, each will move, but because of the length of this, so it will be displacing little more compared to others. Okay, so that is why this B will be correct over here. Okay, it will not move like this, not very far, but yeah, a little bit greater displacement compared to shorter ones. Clear? So this B statement will be correct over here. And A and D is incorrect. Okay, because we already saw the tallest stereocilia involved with depolarization. So it is not like that. It is not that this tall one will depolarize and short one will hyperpolarize. So already we understood why A and D is incorrect. Now let's see the C one. So the hair cells will be depolarized or hyperpolarized in different grades when the axis of displacement is changed. Yes. So when the displacement is occurring in a different axis. Okay. So the depolarization and hyperpolarization, so how much they are moving, right? How much depolarization and how much hyperpolarization is occurring. So that will also change. Okay, because this movement, this axis of displacement is being changed. Clear? So according to this question, according to the statements that have been mentioned over here, B and C will be the right answer. Okay, not just B, B and C both. Clear, Vanishri, Ruben? So the fifth question answer is option 3. Okay, so I have given more information about these hair cells. So they are flask-shaped epithelial cell. Each hair bundle contains around 30 to 100 hexagonally arranged stereocilia. So one taller one, we call it as kinocilium. So they are graded in height and arranged in bilateral symmetrical fashion. So displacement of the hair bundle parallel to this plane towards the tallest stereocilia depolarizes the hair cell, while if they move away, okay, it will cause hyperpolarization. Okay, Vanishri. So let's move ahead. I think I have few more questions. 6. Okay, this is a part B question. Action potential is fired after threshold stimulus, but once the nerve impulse has been generated, there is the interval during which a second action potential cannot be initiated, no matter how large a stimulus is applied. So can you all tell what this gap will be termed as? I just explained you all absolute refractory period, relative refractory period, right? We just saw in some of the questions. Can you all try and tell me? Very good, Reshma, correct. Others try, it is very simple question, part B question they had asked. Right, Vanishri is correct. Yes, Ashwarya right, Ruben, Anupam correct. So like how we already discussed this part, whenever one action potential is generated, okay, another action potential cannot be generated, even though if it is a very large stimulus. So that time period will be, yeah, absolute refractory period. Okay. So it will be something like this. Till this time period, it won't be generated. Okay. After that, it will be a relative refractory period where if it is a very strong stimulus, then there is a possibility that another action potential can be generated. Okay. So that is about absolute refractory period. So what I said, to generate another action potential, what is the requirement for the sodium channels? To generate another action potential, what is the requirement for the sodium channels? What do they have to do? What they have to do if they have to generate another action potential? Resting state. Very good. That is the right word. So the sodium channels have to go back to their resting state. Correct. Vanishri, right. Let's move ahead. So I'm not discussing again about the absolute refractory period. Okay. So it takes about 1 to 2 milliseconds. So time also you should know. Okay. Already I've discussed about absolute refractory period. Now let's move on to the next question. Question number 7. So this is again from sensory organs. The following statements describe the possible functions of outer hair cells of organ of Cottai. So again, this is the question from ears. You can see how they ask you statement-based questions from ears. So the outer hair cells are depolarized or hyperpolarized depending on the direction of movement of stereocilia. The outer hair cells are lengthened in depolarization and shortened in hyperpolarization. C. The outer hair cells decrease the amplitude and clarity of sound by shortening and lengthening. D. The efferent nerve fibers of olivocochlear bundle modulate the sensitivity of the outer hair cells. E. The effect of stimulation of olivocochlear bundle on the outer hair cells is excitatory. So which of the following options, which of the options given below represent the correct statements? So we have already discussed this part. Okay, it is a similar question. Just read carefully and try to answer the right answer, right option. Please tell me. Yes, Reshma was right for the previous question. This is very simple. Okay, Vanishri has already answered. Correct. Absolutely right, Vanishri. It's very simple. Okay, one statement, if you know, you can easily find the right option over here. Okay, Ashwarya, statement A is correct. Very good. So if A is correct, what do you think about B? B is correct or incorrect? I will try to solve your confusion. If A is correct, what do you think about B? A is correct, right, Vanishri? So Ashwarya is getting confused between D and B. So I'm just asking, if you say A is correct, what about B? Will you say B is correct? No, right? Yes, Anupam, it is not correct. So if A is correct, B is not correct. So which one should be the right option? So if you don't know each and every option, it is fine. Okay. So if you know this trick, how to solve these statement based questions, you can easily solve. Okay. You don't have to bother much about other statements, even if you know one or two statements. So now Ashwarya, can you tell me? B is not correct? Correct. Good. So the right answer, students, yes, Reshma, you are right. The right answer will be option 3 over here for 7th question, because just now we discussed just little bit the words, little bit they have made it more longer, so it looks little confusing. So the outer hair cells are depolarized or hyperpolarized depending on the direction of movement of stereocilia. So that is correct, because these are the stereocilia and there's one kinocilium. So if the stereocilia move here, it is depolarization. If they move away, it is hyperpolarization. So they are depolarized or hyperpolarized depending on direction. So A is correct. Now what is B saying? B says these outer hair cells are lengthened in depolarization and shortened. And is it like that? Are the length is increasing? Is this length increasing when they are depolarizing? And is it shortening when they are hyperpolarizing? No, right? Yes, Vanishri, right. So there is just the displacement that is occurring. That the length is still the same. Okay, how much they are being displaced, that is changing, right? So B is not correct over here. So if you know A and B is not correct, means A is correct, B is not correct. So this will be probably the right answer, right? The option 3. Let's see the C statement. The outer hair cells decrease the amplitude and clarity of sound by shortening and lengthening. The outer hair cells decrease the amplitude and clarity of sound. So that is not there, right? They're not shortening and lengthening and they're not affecting the amplitude like that. So C is again incorrect. D says the efferent nerve fibers of olivocochlear bundle modulate the sensitivity of outer hair cells. So here you need to know about this olivocochlear bundle. Okay, so what it is, I'll tell you. So definitely D is correct. So they will modulate, okay, they will play a role in modulating the sensitivity of this outer hair cells. So D is correct. E, the effect of stimulation of olivocochlear bundle is excitatory. So this is again incorrect. So the right statement will be your A and D. Okay, so I'll quickly tell you about this one component of the auditory efferent system. So that component is your olivocochlear bundle. Okay, so what is this auditory efferent system? So the one that afferent takes the information. Efferent will, this is the sensory part, right? And this is the motor part where the effect will happen. So this olivocochlear bundle will play a role over here, which is efferent system. It projects from the superior olivary complex right back to the cochlea. So this is from the brain towards the cochlea. This bundle will reach. it innervates the outer hair cells, means it will reach towards these outer hair cells and it will play a role in amplification mechanism. Okay, so olivocochlear bundle will play a role in amplification mechanism. So that is why it will modulate the sensitivity of this outer hair cells. Okay, the sensitivity, like how much clearly you can hear. So that sensitivity, that modulation is what is the statement D, that is by the olivocochlear bundle. So A and D is the right answer over here. So that was the 7th question. I have one more question. So quickly we'll see this question and then we will try to end. So which one is the correct sequence? So this is again from eyes part. Okay. Now from eyes, usually they will ask you this type of question. Now it can be a part of your unit 7 as well, and the same topic is also taught in signaling also. Okay, the signaling with respect to how vision occurs. So which one is the correct sequence of events that takes place during phototransduction when light falls onto the retina? Okay, so there are 4 statements over here. Try to find it out which will be the correct sequence of events. Okay, closure of sodium channels, activation of transducing, decreased release of, so here there will be a dash, activation of transducing, transducin it is, okay, this G is like this, decreased release of glutamate, decrease in intracellular cyclic GMP, structural changes in rhodopsin. So there are 5 sequence of events that are given. So you have to arrange them and tell which is the correct one. Okay, Reshma, very quick. Yes, correct, you're right. Good. Others try. It is no, it is an important question. If you know this question, you can easily score 4 marks. Ruben, correct. Ashwarya, very good, right? Definitely they'll ask you this type of questions. Okay, every year they ask. Little bit like sometimes they will give you the sequence, sometimes they'll give you the figure to label it like that. Okay, so the right answer for this particular question we will see is answer the option 3. Okay, so what happens when during phototransduction when light falls on the retina? What all sequence of changes happen when light falls on the retina? First, so you have this rods will have rhodopsin. Okay, so this is the, so I'll show you this figure over here. So when light falls on the rhodopsin, so this transducing protein will get activated. Okay, so light stimulation will lead to activation of a G protein. So transducin is a G protein, right? So the alpha part will go and it will activate this activated G part. The alpha will activate the cyclic GMP phosphodiesterase. So PDE is the phosphodiesterase, right? And this is the second step. Now this phosphodiesterase, what it will do, it will hydrolyze the cyclic GMP. Okay, so when cyclic GMP is hydrolyzed, it is removed, so its concentration will decrease. So as a result, now cyclic GMP maintains the sodium channels. Okay, now if cyclic GMP is removed, the sodium will not be able to enter inside. The sodium channels will close. Okay, so this is the sequence of events that takes place. So when sodium channels close, so further, because the sodium channels are closed, sodium will not enter inside, so there will be no depolarization, repolarization, so action potential will not be generated. So because of which glutamate will not be released, and glutamate is an inhibitory neurotransmitter that is being released over here. So when glutamate is not released, so the next cell it will get activated. Okay, so that is the sequence of events that takes place. So first, when light falls, structural changes in rhodopsin occur, transducin gets activated, cyclic GMP level will decrease, sodium channels will close. So decreased release of glutamate. Now, glutamate is an inhibitory neurotransmitter. When its release decreases, the next cell will get activated. Clear? Is question number 8 is clear. So same thing is written over here. I'm not going to read through it. Just understand this figure. Okay, this figure is important. Definitely they'll ask you questions from this figure. So this sequence of events should be clear to everyone. So with this, I come to the end of the session. So just wanted to share with you all this about the Raftar batch, and already I've given you all the details about it, as well as the cloning vector and recombinant DNA hands-on training. Whoever is interested, please register at the earliest so that you can avail the benefits out of it. Thank you everyone, and stay tuned for tomorrow. Again, we'll be having an exciting session where you will discuss more questions, and definitely it is going to help you all in writing your CSIR NET exams. Welcome, Ashwarya. Thank you, students.